Pratima from Planet Physiology. In this video, we will have introduction to blood. You must have heard that the patient has been given blood transfusion for treatment of anemia or for compensating the lost blood volume. What is blood? Blood is nothing but fluid connective tissue, and study of blood disorders and their treatment is known as hematology. Before dealing with composition and functions of each component of blood, we will just see what are the characteristics of blood. As I told, blood is a connective tissue whose matrix is watery or fluid in nature. This blood is confined within cardiovascular system. Arterial blood is scarlet red in color, whereas venous blood is bluish in color. Blood is non-Newtonian fluid. That means it is non-homogeneous fluid. It has got peculiar smell as well as taste. Approximate blood volume is 5 liters and it is about 8% of total body weight. pH of blood is slightly alkaline, it is 7.4. Blood exerts osmotic pressure of 25 millimeters of mercury, and even though it is fluid, it shows property of coagulation or coagulability if it is exposed to any water-wettable substances or collagen. Blood also has suspension stability. Blood is viscous in nature and viscosity of blood is 5 times than that of water, and if you just consider blood without cells, which is called as plasma, its viscosity is 3 times than that of water. Specific gravity of whole blood ranges between 1.052 to 1.062. Plasma is 1.022 to 1.026, and if you just take the cells, its specific gravity is even much more higher. It is 1.095 to 1.099. Now let us study composition of blood. To study blood composition, sample is collected from the body, it is mixed with anticoagulant and then it is centrifuged. After centrifugation, we can clearly note 2 layers. Bottom is red in color, which is nothing but all packed red cells, and topmost is plasma. These 2 layers are separated by one blackish line called as buffy coat. It contains other cellular elements in the blood, mainly the WBCs and platelets. So let us study these components one by one. Because cells are heavier than liquid portion, they settle at the bottom of the test tube. So first we will study the cellular components. All these blood cells are synthesized in bone marrow, and the precursor cell that gives rise to all kinds of blood cells is called as pluripotent hemopoietic stem cell or just as hemopoietic stem cell. The process of formation of these blood cells is known as hemopoiesis. So if it is the process of production of RBCs, it is known as erythropoiesis. Formation of WBCs is known as leucopoiesis, and formation of platelets is known as thrombopoiesis. All these blood cells have got specific lifespan. So once their lifespan is over, these aged cells, or if cells become abnormal, they will be removed by spleen. Now what are the various cell elements in the blood? The most abundant cell element in blood are red blood cells or red blood corpuscles, which is abbreviated as RBCs. The word erythrocyte is synonym for RBCs, and this has 2 terms in this, erythros and cyte. Cyte means cell and erythros is red, so red cells. Next important cells in the blood are called as white blood cells or leucocytes. Here again, leukos means white and cyte means cells, so white cells. The third component of the blood cells are platelets. These are very small, minute, disc-like cells, and therefore they are called as platelets, and they are also called as thrombocytes because they play important role in thrombus formation or clot formation. Let us study these cells one by one. Erythrocytes are biconcave non-nucleated cells and they contain protein called hemoglobin. This hemoglobin is basically iron containing compound, and because of its iron, it imparts red color to the cells and therefore they appear red in color and hence named as erythrocytes or RBCs. Their count in the blood ranges between 4.5 to 5.5 millions per cubic millimeter, and this count will be little different in males and females. The main function of this erythrocyte is to transport gases. Hemoglobin within the RBC will help in transport of oxygen from lungs to the tissues, and one molecule of hemoglobin can carry 4 molecules of oxygen. These RBCs will also carry carbon dioxide from tissues to lungs. Another important function of RBC is acid-base balance, because hemoglobin can act as buffer, so it will prevent changes in the pH of blood. RBC membranes have specific protein moieties which are responsible for imparting a particular blood group to the person. RBCs also make blood viscous. Coming to the second type of cells, leukocytes or WBCs. These leukocytes can be divided into two, granulocytes and agranulocytes. Granulocytes are of three types: neutrophils, eosinophils and basophils. Agranulocytes are of two types: lymphocytes and monocytes. Leukocyte count ranges between 4,000 to 11,000 cells per cubic millimeter, and all the leukocytes are nucleated cells. To remember different types of leukocyte, here is a table. In case of granulocyte, as name suggests, their cytoplasm will contain granules. So, granulocytes will possess granules, but agranulocyte will not. Nucleus of granulocyte always will be lobed. Routinely 2 to 5 lobes of the nucleus will be present in granulocytes, but the nucleus in agranulocytes will be either unlobed, single nucleus, or it may show slight indentation. Granulocytes are classified based on the staining property of the granules, and therefore these cells have suffix phil. Philia means affinity, attraction. So, first is neutrophil. The granules of neutrophils will be stained by both acidic as well as basic dyes, and therefore these cells are called as neutrophils. Similarly, granules of eosinophils will be stained by acidic dyes like eosin. So, eosinophil. Basophil granules will have affinity towards basic dyes like methylene blue, hematoxylin, so they are known as basophils. In contrast, agranulocytes will have suffix in their name as cyte, lymphocyte, monocyte. Main function of leukocytes is defense of body against various pathogens like viruses, bacteria, parasites, carcinogens, etc. Neutrophils and monocytes will defense by the process called as phagocytosis. Eosinophils are mainly working against parasites. They are also anti-allergic in nature. They will prevent allergic reactions. Lymphocytes confer immunity, whereas basophils are mainly concerned with allergic reactions. The last cellular component is thrombocyte. These are small disc-shaped non-nucleated cells. Their count ranges between 2 to 5 lakhs per cubic millimeter, and they play important role in hemostasis, that means prevention of blood loss from injury. This is achieved by vasoconstriction, platelet plug formation, blood clotting as well as clot retraction process. So, here we finish with the cellular portion. Let us move on to the liquid part of the blood, that is plasma. Plasma constitutes 55%. It comes to about 3.5 liters, and normally it is light yellow or straw color liquid. 91% of the plasma is water and remaining 9% is solid. Solid components we can divide into two, inorganic and organic. Inorganic components constitute just 1% and it will be in the form of electrolytes as well as gases. Organic components constitute 8% and out of that 7% is by proteins. So, these proteins are collectively called as plasma proteins, and 1% will be other organic constituents like nutrients, glucose, fatty acids, amino acids as well as metabolic waste like urea, uric acid, creatine, etc. So coming to the plasma proteins, there are three important proteins in the plasma: albumin, globulin and fibrinogen. All these plasma proteins in case of embryo are formed from mesenchymal cells, whereas in adults the major source of all the plasma proteins is liver. Some proteins come from lymphocytes and macrophages, and about 6 to 8.5 gram per deciliter plasma proteins are present in the blood. These plasma proteins can be identified as well as separated by various processes like precipitation by salts, fractional precipitation, ultracentrifugation as well as electrophoresis. Let us see about albumin. Albumin is a small globular protein and it has negative charge on its surface. Its molecular weight is 69,000 and it is synthesized from liver. Normal value of albumin is 3.5 to 5 gram per deciliter. Albumin concentration can be physiologically decreased in case of neonates as well as during pregnancy. Pathologically, it will be decreased in case of liver diseases where synthesis will be a problem, in renal diseases where it will be lost in urine, and in case of burns where it will be leaked out from the circulation. This albumin is mainly responsible for exerting colloid osmotic pressure because it is osmotically active particles, means it will tend to attract water molecules towards itself, and this is very important to prevent loss of water from the capillaries. It also helps in preventing edema. So this 25 millimeters of mercury, whatever is the colloidal osmotic pressure, it is mainly exerted by the albumin. Albumin also acts as carrier protein for various hormones, amino acids, fatty acids, steroids, bilirubin as well as for certain drugs. Next plasma protein is globulin, and as name suggests, it is globular in structure. Molecular weight of globulin will vary depending upon the type of globulin. For example, alpha 1 globulin molecular weight is 2 lakhs, whereas beta globulin molecular weight is 90,000. Globulins are synthesized from plasma cells as well as reticuloendothelial cells. Normal value of globulin ranges between 2.5 to 3.5 gram per cent. Globulin concentration can increase in liver diseases, multiple myeloma, acute nephritis, leukemia and tuberculosis. Let us study functions of globulin. There are different kinds of globulin, so we will see individual functions. Alpha 1 globulin mainly helps in transport of lipoproteins, and it also acts as protease inhibitor, and the protease inhibitor of alpha 1 variety is called alpha 1 antitrypsin. Alpha 2 globulin helps to transport substances like copper and hemoglobin, and it also acts as protease inhibitor. An example is alpha 2 macroglobulin. Beta globulins are concerned with transport of iron, thyroid hormones and cortisol, and they are also important components of coagulation system, fibrinolytic system as well as complement system in the blood. Gamma globulins, they act as immunoglobulins or antibodies. There are 5 types of gamma globulins, namely IgA. Ig stands for immunoglobulin. So IgA, IgG, IgM, IgE and IgD. All these antibodies, they play important role in defense against invading organisms. Let us study clinical aspects of this albumin and globulin. So even though there are conditions where albumin production will be reduced or albumin levels will fall or globulin levels will rise, but what is the most important thing is the albumin to globulin ratio, and this ratio can be altered in various diseases. Like normal ratio is 1.7:1, and it can be altered if the protein intake is low, as in case of protein energy malnutrition, or if there is decreased synthesis of especially albumin, say in case of liver diseases, or there is excess of protein loss. Again, this albumin is the main protein which can be lost easily from the circulation in cases of burns or in renal diseases. The third type of plasma protein is fibrinogen, which is also called as clotting factor 1. This is elongated fibrous protein and its molecular weight ranges between 3.5 to 4.5 lakhs. It is synthesized from liver, but its concentration is quite low. It is just 0.2 to 0.4 gram %. Because fibrinogen is elongated molecule, it is responsible for offering viscosity to the blood. So, whatever is the viscosity of the plasma, it is mainly because of presence of fibrinogen, whereas viscosity of the entire blood is because of RBCs as well as fibrinogen. This fibrinogen is also the important part of coagulation system of the blood because it is clotting factor number 1, and it also plays role in rouleau formation, that is piling up of RBCs if anticoagulated blood is allowed to settle. This fibrinogen concentration can increase during conditions like pregnancy, menstruation, smoking, tissue injury, acute infections as well as malaria, and its levels can decrease in case of liver diseases. Now, let us study cumulative functions of plasma proteins. First thing, it is the maintenance of colloidal osmotic pressure, and it is mainly because of albumin. Plasma proteins play important role in blood coagulation, and this is because of fibrinogen as well as various types of globulins. And maintenance of viscosity of blood is because of fibrinogen. Maintenance of acid-base balance, it is because of mainly albumin, because it is the most abundant protein in the blood. Then immune function because of gamma globulins. Transport function because of albumin as well as globulin. Plasma proteins, they act as reservoir, so they can have these are the extra proteins which can be utilized in case of emergencies, and again in case of emergencies they can provide nutrition to the body, but this last function will be seen only in extreme cases of starvation where there is no energy source available. Only in that case plasma proteins can be broken down to provide ATP, otherwise not. Then you must have come across the word serum. The serum is nothing but plasma without clotting factors. Blood sample is taken out and it is just left as it is allowed to clot. Within 20 to 30 minutes blood sample will clot and that clot will retract. After clot retraction, the fluid will oozed out and this fluid is called as now serum. So, because blood is clotted, all the clotting factors in that blood sample will be utilized, and that is the reason serum will be devoid of any clotting factors. Now, this serum can be utilized to check levels of electrolytes, albumin, urea, uric acid, creatinine. Many investigations can be performed by using serum. Now, let us study functions of blood. We have finished with the cellular component, we have finished with the liquid component. Now, together they are forming blood, and so we will study now functions of blood. The important function is respiratory function, that is transport of gases, respiratory gases like oxygen as well as carbon dioxide. The nutritive function is because blood is the means by which nutrients are transported to various tissues like glucose, fatty acids. Blood is also the means by which excretory products will be distributed to various excretory organs like metabolic waste products like lactic acid, carbon dioxide. Blood plays vital role in homeostasis. What is homeostasis? It is the maintenance of constant internal environment. If you want to know details about homeostasis, you can refer to my previous video on homeostasis. So, what is the role of blood in homeostasis? It will help in regulation of water electrolyte balance because blood volume reflects changes in the extracellular fluid volume. Then it plays important role in acid-base balance to regulate pH of the blood. Another important homeostatic mechanism achieved because of blood is temperature regulation. Next function is protective function because of presence of WBCs as well as immunoglobulins. Blood acts as transport medium for nutrients, hormones, metabolic waste products, drugs, etc. Coagulation is another important function of blood mainly because of fibrinogen and other globulins. RBCs as well as fibrinogens are responsible for imparting viscosity to the blood and which is playing important role in various hemodynamic principles. Albumin as well as electrolytes like sodium, they play important role in maintaining plasma colloidal osmotic pressure, which is highly essential to prevent edema. Now let us study some clinical aspects related to blood. Whole blood can be transfused to the patient to replace his lost blood volume. Only packed cells also can be given, where only RBCs can be given to the patient if the patient's condition do not support the entire blood transfusion. Similarly, in diseases like dengue, you can give platelets only. Various components of the blood can be separated and they can be stored for later use for many months together. For example, we can have fresh frozen plasma as well as clotting factor as well as some antibodies separated out which can be used in treatment. Also, blood can be used for various diagnostic uses. We can check RBC count, hemoglobin, WBC count, etc. for confirmation of various diseases. We can check blood sugar level, blood urea level. So, all these tests which can be done on small blood sample can be very important in diagnostic purposes. So, this is all about introduction to blood. Thank you. If you enjoy my sessions, press the like button and share it with your friends. If you haven't yet subscribed my channel, press the subscribe button. To get notifications about new releases, press bell icon. Thank you for watching and see you in the next video.